off you go.
Thank you, Mayank. Thank you for inviting me, and also to Greg for inviting me. I'm sorry I can't stay longer here on a flying visit. Mayank asked me to give a kind of a general talk to give you the background, overall framework, which will set the situation for the next couple of weeks. So I'll do that, and I'll do it in a quasi-historical way. As we all know, history is told by by certain people, and there are very different versions of history. So I'll tell you my version of history, and then I'll tell you more specifically about our ideas that the place cells, particularly the place cells in the hippocampal cognitive map, really receive two independent inputs. And this was something which we suggested a long time ago, and I think we're now pretty close to having pretty reliable and agreed upon evidence that this is the case. So let me start then where all ideas about the hippocampus really begin. And that's with the study of Henry Mollison, who in 1953 received bilateral temporal lesions in the mesial temporal lobes here. Here's shown that the deficits, they're bilateral, so he lost all of this tissue on both sides. It's a surgical operation. Tissue is removed in a radical exploratory operation to try to alleviate his epilepsy. There was good reason to believe that the foci for the epilepsy were in the temporal lobes. The operation was a success. The number of seizures were reduced, but unfortunately, Henry was left for the rest of his life with a very, very profound and global amnesia. That means he could not acquire new information, and also that he lost a considerable amount of memories and information that he had acquired before the operation. For a long time now, there's been a controversy as to exactly what the information was that HM had lost, particularly what's called the retrograde amnesia. But now, after many, many years of studying him, originally he was studied by Brenda Milner, and more recently by her student Suzanne Corkin at MIT, who's now published a fairly extensive book on her experiments and other experiments on HM. What they've concluded is that he's essentially cannot recall anything that relies on personal experience. He's lost what we would call episodic or autobiographical memories. Okay, so his recollection from the time before his operation was sketchier than initially believed. In fact, it looks as though he has a permanent loss of memories of the autobiographical time before the operation, such as a specific Christmas gift his father had given him. He retained only the gist of personally experienced events, plain facts that no recollection of specific episodes. His deficit was specific to his autobiographical memory. So what we mean by autobiographical memory, then, is the memory for events which occurred in a specific time and place. And I think that's important, because if the memory loss he had was much more extensive, then it would be very difficult for us to model it in animals. But in fact, if it is in fact a memory loss for events in specific times and space, then we have a hope of actually modeling it in animals. Let me just introduce you to the hippocampus, for those of you who are not familiar. The hippocampus is, as I showed you already, in the medial temporal lobe, sitting here, has two major inputs to it and two major outputs. One major input to it comes from this area here called the septum, which is connected via this very large fiber bundle, the fornix, one of the largest fiber bundles in the forebrain, to the hippocampus, and conveys to it all sorts of information about body states, information about the animal's movements, and also takes out of the hippocampus information which can be used to control aspects of hypothalamic and brainstem function. And one of the things I'll talk about tomorrow is the way in which that input, and particularly the cholinergic and GABAergic inputs coming in from that pathway, are responsible for creating the hippocampal theta rhythm, one of the important oscillatory activities in the hippocampus, which we think is an important clue to how it carries out some of its functions. The other prominent input to the hippocampus comes from neocortex. This is taken from Larry Squire. It's a very, very simplified version of the cortical inputs to the hippocampus. I'll show you a much more complicated one in a second and show you why we have great problems in coming up with animal models of hippocampal function. But essentially what Squire's point is that if you take the activity in cortical association areas, visual, auditory, and areas like that, and follow their pathway, they impinge upon these areas which are actually surrounding the hippocampus, and part of what we would call the parahippocampal regions. So you see here, many inputs are coming into this area here called the entorhinal cortex, either directly or indirectly, which is one of the major portals into the hippocampus. So the hippocampus has two major inputs. It has inputs about information about the sensory world, the sensory environment, and then it has information which is a much more modulatory information coming in from the septal regions, which is controlling the way in which that information is put together and modified in the hippocampus. Now, this is a very simplified version of the anatomy of the hippocampus, and I want to show you a less simplified one. Because this is the famous Van Essen visual hierarchy model, in which David Van Essen tried to see how you would follow the analysis of visual signals from the retinal ganglion cells through the lateral geniculate and then up through the cortical pathways that end up finally in this area of the hippocampus up here. And as you can see, it's a nightmare. So the program of Ublin-Wiesel and the program that's being followed by most cortical physiologists and most cortical neuroscientists of moving from the periphery and going through a series of way stations through one of these two pathways, the ventral or the dorsal or the ventral stream, is doable, but it's probably going to take centuries before we actually are able to get it to work. There are just too many interactions to sort of treat this as a serial system. So having that in mind, a long time ago, we thought, well, if we're going to come up with some idea of what the hippocampus is going to do in animals and get some insight into how it could be a model for human memory and human memory deficits, perhaps we should just bite the bullet. And instead of sort of working our way through this, why not just go and look at the hippocampus itself? Why not just stick electrodes into the hippocampus and see what the cells are doing? What are the cells telling us? Now, at the time, and I'm talking now about the early 70s, most people thought this was loony. They thought this was crazy. They thought the idea that you could actually take a system which is getting, and this is just the visual system, information of all this complexity pouring into the hippocampus and make some sense of it was just not going to be possible. On the other hand, if you think about the brain as the interface between the physical world and the mind, and that we essentially subjectively and introspectively have access to the mind, we have all kinds of ideas about that are going on in our mind about space and time and objects and all these sorts of things, which I guess all the physicists out there would just tell us these things don't exist in the physical world. I mean, that's not what's out there, folks. Then you might end up seeing things at this end of the spectrum, at this end of the anatomical hierarchy, which are easier to interpret than things that are down here, which are much closer to the physical world of quantum mechanics and things like that. So with that kind of idea, we just stuck electrodes into the hippocampus of rats, let them recover, and saw what we saw. And the reason that it's reasonable to do that is because there's a lot of conservation of hippocampal anatomy, structure, and we assume and we assume function, up the phylogenetic scale. So here's the hippocampus in the human, and that's the area which was removed in HM. And here's the hippocampus in the rodents. They're, as I'll show you in a second, in cross-section, they're essentially the same. They've just moved around a bit. So whereas the hippocampus is lying here in the parietal and temporal regions of the brain, here it's been pulled around and lies entirely in the temporal regions in the human. If you look at the cross-section, though, see this very nice, familiar structure, which essentially consists of two interlocking sections of cell bodies, the CA1, CA2, CA3 cell bodies here in the dentate gyrus there. And you see that same thing recapitulated in monkeys, and you see the same thing recapitulated in humans. So it's reasonable to think that if you found out something about what was going on in the hippocampus of the rat, that would perhaps inform your ideas about human autobiographical human episodic memory. And that's what we did. So I won't go on to this. What we did was we put electrodes into this part of the hippocampus here. You can see all of those cell bodies are really nicely lined up. They're pyramidal cells, so all their apical dendrites are running off parallel to each other. It makes a beautiful anatomical structure. I won't go into much more of the anatomy, but all the inputs, for example, to this area come in as fibers passing en passage along many individual dendrites. It's a kind of a computational neuroscientist ideal structure to look at. It gives you lots and lots of scope for all kinds of associative processes. So we put electrodes in here, not to ignore the fact that here's the hippocampus proper, with its CA field, but that there are lots and lots of other areas behind the hippocampus, the parahippocampal gyrus here, which are very tightly interconnected anatomically with the hippocampus, and including this subicular area here, composed of the spiculum, presubiculum, parasubiculum, and the entorhinal cortex, which is that area where all the sensory information from the neocortex is funneling in. And if you look at the anatomy of these, I won't go through this. This is a schematic of it. There are inputs coming into the hippocampus from the entorhinal cortex, and then there are inputs going back out. It forms a closed loop, and you can get lots of possibilities for interactions of that sort. Okay, so what we did was we placed electrodes, as I say, into the hippocampus, and just let the animals go about their daily business. And it took us quite a while to make any sense of it, partly for the following reason, that the cells are essentially quiet. They're essentially turned off for most of the time that the animal is going about its daily business. So if you put an animal on a platform like this and let it wander around, get it to press a bar to get some food, get it to approach things, get it to run away from things, record from the cells when the animal's sleeping, when it's grooming, doing all sorts of things that an animal normally does, the cells really are quiet most of the time. Every once in a while they'll show a burst of spikes, you know they're there, but essentially they're quiet. And it's only when we began to realize that the important variable that was causing the cells to fire in a consistent manner was the animal's location in the environment, and not particularly what the animal was doing, that we began to make any sense of what the system was doing. So this is the first really good example we had. Here's a cell which fires when the animal comes over here, and not when it goes to lots of different parts of it. And here's the raster plot, the histogram plot, and you can see that the cell only fires when the animal's over here or here. Here are the spikes. And on the basis of this, we began to think, well, maybe this is providing the animal, a rat now, with a spatial reference mapping system. And we suggested this finding suggests that the hippocampus provides the rest of the brain with a spatial reference map. And that recalled some of the ideas that had been put out there many, many years before by E.C. Tolman in Berkeley here, who had said that animals do have some sort of map in their brain, and he used that idea to try to explain some effects and some behaviors that he had observed in maze learning. Yeah?
Does the area under the histogram correspond to how often the rat visited location A, B, C, D?
No. So in this case, in this case, he's going to various places. So yes, you're right. It doesn't control for the fact that he's visiting them for different amounts of time. Of course, now subsequently we take that into account, as I'll show you in a second. So this is just the first observation, and you're right. You have to sort of trust me that I get him to go to the different places equally often. Yeah? Is there another question? Okay. So. So what Tolman had said is that we believe that in the course of learning, something like a field map of the environment gets established. And this was in contrast to stimulus response ideas, which had been put forward by Clark Hall at Yale, and which really dominated the field, certainly into the 60s and 70s when we first started these experiments. The question of how animals found their way around the world and how they solved maze problems was more or less resolved, I think. I think most people thought that they did it the way Hull thought, that an animal solved the maze by running to the choice point and then making a left turn at that choice point and then running to the next choice point and making a left or right, associating each stimulus configuration at the choice points with a behavioral response. And the idea that Tolman came up with, that instead of this, the incoming impulses are worked over into a tentative cognitive-like map of the environment, really didn't have much purchase. It didn't have much going for it. But he said, this is, if you had a map like this, it would enable you to do a lot of things, in particular detours and umwegs that they had observed in rat behavior. It's only when we could go and look into the brain and look at how different bits of the brain were working that we could actually begin to resolve the issue between these two very, very radically different views of the way that the system was working. So what we suggested, and particularly with Lynn Nadell, who joined us at around that time and with whom we wrote a book called The Hippocampus as a Cognitive Map, it's a few years ago now, was that if you had a whole bunch of these place representations, you'd have the beginnings of a map-like structure, a map-like system. Just having a bunch of place representations doesn't give you a map, because what you need in addition to having the place-like representations, A, B, and C, was some way of connecting them together. So that if an animal were in place A and wanted to go to place B, it would know what the vector was which connected the two places. Or if it were going from a long vector AB, it would know that it was going to end up in place B, that is, and would be able to call up before it got to place B. The system would be able to expect the stimulus configuration that would occur in place B. Around that time, Richard Morris decided he was going to stop being a stimulus response theorist and behaviorist, and invented a task which was designed to test this basic idea, the water maze task, in which he said, well, what if you're right, this is now in the late 70s, early 80s, what we would expect is that animals without this part of the brain would be able to, not be able to do the sorts of navigational types of behaviors or engage in the navigational behaviors that we know that normal animals can do. So he came up with a task which was a very simple task. You put a platform in a pool here, and it's hidden so the animal can't see it. Here's the platform. And then you put the animal down in this water, and after having a leisurely swim around, he decides he's had enough of this and he wants to get out of the water. So he goes and he jumps onto the platform, and then you remove him from the pool. It's a very simple navigational task. And what you find is that when you first put him down here, of course he has no idea where the platform is, but after a few trials, and it is very few trials, this is the 8th trial, the normal animal runs directly to the platform, showing that he has an idea where the platform is relative to the cues. If you make lesions in the hippocampus of animals like this, then they continue to show this kind of behavior for as long as you test them. Okay, I want to go back to this idea then that you can produce this map-like system by having a series of place cells connected together by these vectors. One of the things we thought about right from the beginning is that it's quite likely that each of these place cells receives two different inputs. There's the obvious one, which is that the animal and the cells can tell where the animal is by doing a simple assessment of where the various cues in the room are, and doing a kind of triangulation, and using sensory or environmental stimuli to assess where the animal is, or the person is. On the other hand, there's another possible way of finding out where you are, and that is once you know where you are in one location in an environment, you can update the representation of where you are by using inertial navigation, or what we now call path integration information, based on your own bodily movements to update the representation. So what we suggested is, not unlike the way aircraft find their way around the world, is that information about changes in position and direction in space could be calculated from the animal's movements, and that could be used to update the representation. So once the system knows where the animal is, it can then use this internal system, irrespective of information coming in from the external world, to update the representation. The hippocampus could calculate subsequent positions in the environment on the basis of how far and in what direction the animal had moved in the interim. And that's it's that suggestion which I want to sort of talk about in the second part of the talk. And what I had in mind here is, this is a kind of a way of making that a little bit more explicit. Using this famous painting, The Flagellation of Christ by Piero della Francesca. He's a 15th century Tuscan painter, who in fact was the first one to actually get all the mathematics of projective geometry correct, so that you get a true representation of three-dimensional spaces on the picture plane. And what he's showing here is three Tuscan gentlemen conferring about the weather, or perhaps the state of the economy in Milan. And in the background here, shown in this very nice three-dimensional representation, is the flagellation of Christ during his crucifixion. And what we were suggesting, or what I'm suggesting now, is that if we take this chap here, he can localize his position in this environment in two different ways. He can, for example, tell where he is by noticing where the various landmarks are, the pillar that Christ is attached to, this other pillar here, this very nice house in the background here. And he could also do that for when he goes over to this location here. So any place that he finds himself, as long as he has access to these environmental stimuli, he can tell where he is. On the other hand, once having localized himself there, he can find his way to this location and also tell where he is when he's at that location by noticing that to get there, he has to rotate by 180 degrees and then walk a certain number of steps, or if he has a better metric, a certain number of meters or feet, turn right 90 degrees, and then walk another second. So that independent of those cues in the environment, he could localize this location using this internal path integration system. Okay, well, what I want to tell you now is that we, over many, many years, in not so much in our lab, I have to say, I'll explain exactly what the process was, but in many labs around the world, there have been considerable success in identifying all of the different aspects of the navigational system. It turns out that they're not in the hippocampus proper. Having thought about all these things and said, well, where is this information? I stuck in the hippocampus and kept recording there rather grudgingly, whereas other people went and looked in other parts of the hippocampal formation and found these other cells. Techniques we use are shown here. We place, and by and large, they're all the same techniques that I'll talk about. We place electrodes into parts of the system, in this case the CA1 fields here. Place the animal in a very, very simple environment, such as a square or a circular box. Let the animal recover from the surgery. See where the cells are firing as it moves around in this environment. And we know where it is because we have a camera that's tracking diodes on the top of its head. So we know where the animal is at any time, going back to your question before. And so it can take the actual location of the animal and the statistics of its location into account. And we also know where the animal is when the cells are firing. And what we typically see is a more modern version of the picture I showed you earlier, that as the animal runs around in this environment, you see that it covers the environment fairly equitably, and we can, and also if we look at where the cells fire, we see that they tend to fire in one localized position. In this case, the cell fires in the southwest corner. And we can show that as a heat map where the red colors show the highest firing, and successively cooler colors show lesser firing. So this is a cell which fires at 10 hertz in the center of the field, but not in any amount of firing elsewhere. Okay, and what I briefly want to tell you then over the next 15, 20 minutes is that we now know that in addition to the place cells, which are found in the hippocampus proper, there are several other kinds of cell types which essentially provide us with the information that we need in order to provide those two different types of information to the hippocampus. We know that there are cells which respond to landmarks in the environment, which are called boundary vector cells. And we also know that there are cells which respond to the animal's heading direction, providing that heading direction information. And the final class of cells are cells, the grid cells, which we think are providing something like a metric for the spatial map. So I'll tell you about those different cell types briefly, and then tell you about our experiments, which I think show pretty definitively that there are, in fact, these two independent sources of information for any given cell.
Will you tell us how a naive rat, who for the first time is placed, so he doesn't yet have any place cells, how he builds up the firing of the place cells in the southwest corner? He must probably go around a little bit before that.
So that's actually, yes, that's a good question, and it looks as though some of the cells are there almost right from the beginning. Certainly some cell types, they're there almost as soon as you can record them. Part of the problem with the place cells is that the rates are in general low, so that it's very hard to know exactly where the cell is, the cell field is, until you've had enough, the animal's had enough experience. But insofar as you can do that, and then, as it were, find out where the field is, and then look back to see whether the cells are. There are some cells which then fire almost immediately the animal is put into the environment. Other cells do look as though they build up their firing fields over a short period of time, in a matter of minutes. Once the cells have actually had their fields in an environment, they tend to keep them and fire in the same place over successive times in the environment, as long as you don't make changes in the environment. Are there any other questions? Yeah.
So I guess you could test the fact that these two mechanisms exist by sort of turning off the light. You know, you put a rat somewhere, it learns the environment, learns certain cues from the environment, then you turn off the light and you see whether it can still navigate by doing this path integration?
Yeah. So that's correct, and we and others did that right from the beginning. The problem is that assumes that you know that the only environmental sensory cues are visual cues. And we have no reason.
I mean, of course, we'd have
so the fact when you do that, what you find is an animal that's in a familiar environment. When you put it into that environment and then turn the lights off, the cells continue to fire in the right place. The question is that because there are other inputs,
Right.
such as olfactory inputs, auditory inputs, and
Yeah,
things like
yeah.
that. So you need to have a much better control over the sensory environment. I'll show you experiments
Okay.
in which we've done that.
Okay.
Okay, so let me just show you an example of a cartoon of a place cell, if I can get this to work. So here's real data. It's a cartoon, but it shows you the audio is not working. Did the audio not get? Okay.
I don't know.
Yeah. Okay. Well, anyway, you can see that there's supposed to be clicks which represent this. You can see as the animal runs around, it's moving in all directions, it's facing in all directions, and what you see is that the cell only fires in this circumscribed area, and that's shown in this heat map here. Oh, there you go.
Is that it?
Yeah, that's better.
Do you want to
Great.
start that again?
No, I think that's okay. Fine. So as you can see, that that cell only had one field. If you record from lots of cells at the same time, what you see is that they each have their own area, their own localized place field, as they're called. But they're different. And if you record from 30 or so odd cells, what you find is that they each have a different one. And if you line them up in a representation which puts the cells which fire up in the northeast corner here and the northwest corner here and the southeast corner here, what you see is that they more or less represent the entire environment. This is 30-some odd cells out of several 100,000. So there's a multiple re-representation of the environment across the hippocampus. And you can also see that as the animal moves, they more or less cover the entire space here. So as the animal moves from this location here in the northwest corner along this north side of the box here, the cells, there's always a cell representing its location. And similarly, as it moves down this west wall here and along the south.
What fraction of cells are place cells?
So that's a good question. Let me answer it after the next one. If you look at any given environment, it depends to some extent on something like the complexity of the environment. It's not at all clear exactly what the variable is, but it turns out it's something like 25 to 40% of the cells that one records, and you know you're recording from, have fields in that environment. But if you now begin to take the same group of cells and record from different environments, then what you find is that more and more of the cells have place fields over the across the sum total of the environments. And that's shown here. So if you record in two environments, these are environments which the animal is very familiar with. They're very similar environments, and it takes a while for the animal to learn to differentiate between these environments, but this is quite representative of two environments. You record in a circle and a square, what you find is that the cells, some of the cells tend to fire in the circle but not the square. Okay? Some of the cells fire in the circle but not the square and not the circle. And some fire in both environments but differentiate between them on the basis of where they fire in that environment. So you can't tell where the animal is on the basis of any single on the basis of any single firing pattern. It's a distributed code, and by doing experiments in which people have recorded from a whole series of environments, what you find is that it probably is the case that all of these cells have place fields in at least one of the environment. Many of them have fields in lots of the environment.
Have you tried experiments where the rat moves in 3 dimensions, where you can go up and down and tunnels?
So I don't want to steal Kate's. I don't want to steal Kate's thunder here. She's going to tell you all about 3 dimensions in her talk tomorrow. Yeah.
Do you have any understanding of the length scale of these spots? Is it a constant fraction of the size of the environment? Does it vary with the complexity of the neighborhood? If you thought of some kind of archiving code, like you said,
It's a good
Adapt
question.
to the complexities of
So there's, to some extent, it depends on how familiar the animal was with the environment. These are about a meter in diameter. So these are about 30 centimeter fields. As I'll show you later, in one of the experiments we did, we changed the aspect ratio of the environment. And in animals, if you do it the right way, then you can see that the field stretches not completely to maintain the right proportion of the environment, but it does have the shape of the environment and the size of the environment does have an effect. And if you make the environments big enough, then sometimes you begin to see multiple fields.
You begin to see
Multiple fields. You see several fields. Oh, there it is. So this is an experiment that Neil Burgess and I did some years ago. This is only a very small part of it, in which we recorded from cells and then looked at what happens when you change aspects of the box in which the animal is foraging. And of course, these animals are just foraging around. They're not really supposed to be paying any attention to the environment. They're just foraging around, trying to find food that's randomly thrown into the environment. So what we did here was we recorded we trained them in this square shape, and then we distorted it by doubling one or the other of the dimensions, or doubling both of them. So we essentially recorded in 4 boxes, large and small square, and then 2 rectangles with the sides of the squares. And here's a particularly clear example. As you stretch the box in this dimension, you can see this cell actually being pulled out to reflect that change. Some of the changes are a little bit more subtle and not so obvious, but this suggested to us that, in fact, one of the things that was causing the cells to fire in a particular location was the animal's location relative to the walls of the box. The walls of the box were actually acting as landmarks. And I won't go into it too much, but
not all cells do that, right?
Hey?
Not all cells do that. Some do, some don't.
It depends to some extent on the environment, but not all of them do, and some of them do it in different ways. But it's an existence proof. The fact that some of them do that suggests that there are cells. Not all of them do it, but so you have to have a model in which you can say why some of them don't do it.
What fraction is it about?
So some of them just stay nicely here, right? So some of them just stay right over there. And now you've got me. What's the fraction? The fraction which do what? Which show this stretching effect?
Yeah.
Quite a few of them. In this experiment,
Like
there's quite a few.
half-ish?
No, it's about a third. But it does suggest, and so you need a model which suggests why you would get it in some cells and not others.
2/3 pretty much stay the same size. Maybe move around a little bit.
Yeah, or some of them, as you change it, some of them just disappear. So some of them not only show this, but they also show that they are responding to, or some of them change their firing rate. They're certainly showing that they're reflecting the boundaries or borders of the environment. Okay, well, to try to capture all of that, Tom Hartley, Neil Burgess, and I suggested that maybe what these cells are getting is environmental input. And they're getting inputs from some kind of cell, which we call boundary vector cells. I think probably boundary cells is a better name now, which actually was representing the animal's relationship to one of the walls in the environment. And specifically, its distance to the wall in the environment, and it had to be a wall in a specific direction. Because of that previous experiment, I manipulated and changed all the walls so that there couldn't be any specific wall information. So I would swap all the walls around to make sure that it wasn't some smell on the wall or something like that. So it's a wall in a particular direction. And if you had cells like this, for example, here's an example of one which, as the animal got closer to the wall, the firing rate would increase. And you had ones which had different preferred directions in which they were cared about the walls. Then you could put them together in a fairly simple integrate and fire type of system, in which this cell would fire in different environments in the following way. It would always fire towards the north wall. This one fired towards the east wall. And if you added them together, you would get something that began to look like the place cells, purely on the basis of environmental inputs. And sometime after we had made this suggestion, Colin Leave in our lab went and looked not in the hippocampus, but in the subiculum, and found cells which look essentially like that. They fire with these long slabs of firing rates, which are related to one of these walls. So this one we could imagine is related to the west wall, and this one is related to the south wall. So we think those are the cells which are actually providing the sensory information to the place cells, the environmental information. And you can see if you add two of them, which are, say, at right angles to each other, they'll produce something like a rudimentary place cell. Around 1970, I want to tell you now about the other kinds of information. Around 1984, Jim Ronk, working downstate in Brooklyn, reported the existence of cells which were providing information about the heading direction of the animal. So these are cells which don't care about where the animal is in the environment, but care specifically about the direction in which his head is pointing. Here's an example of one, I hope. And what this is showing, again, it's a cartoon made from real data. This is dropping arrows off whenever the cells fire. And you can see that it doesn't really matter too much where the animal is. The arrows are being dropped when he's roughly pointing in that direction. Here's the summed vector of all of these vectors. And in the end, this is going to turn into a representation in a polar plot system like this, showing the average heading vector of the cell. So these are cells which care not so much about where the animal is, but which direction its head is pointing as it goes around the environment. And the interesting, these cells are found not again in the hippocampus, but in the presubiculum here, again in an area which is providing information to this general area behind the hippocampal system here, and eventually providing information into the hippocampus. The interesting thing about these cells is that if you look at two of them or more, and look at their relative preferred heading directions, what you find is that those preferred heading directions, the relationship between those preferred heading directions stays constant. So, for example, if you find a cell which refers in a particular environment under certain circumstances to fire when the animal is pointing to the north, and another one which is pointing to the northeast, and another one to the southeast, and then you do some manipulation which changes that relationship to the relationship of these, for example, spinning it around and getting it dizzy, to the external world, what you find is that all of these internal relationships remain consistent relative to each other. So if you rotate this whole, you find that this one is rotated by 90 degrees, you find that all of the rest of them are rotated by a similar amount. And I think Kechen is going to tell you all about how you can build models to set up networks which will do just this. And then finally, the last type of cell is the most recently discovered one in the Moses lab in Trondheim in 2005, which is a cell that we think provides something like the metric for the cognitive map. And these are the grid cells. And what they found is that there are cells in the entorhinal cortex. I can put that up here. Another of these parahippocampal regions, which have not just a single firing field, but a whole bunch of firing fields, which are laid out in a very, very regular hexagonal, or if you prefer, triangular pattern, so that the peaks of all of these fields form a consistent pattern across all of the environments that the animal finds itself in, and which look like they might be providing something like the Cartesian metric for the cognitive map. So when you see one of these cells, initially it doesn't seem to be providing any spatial information to the rest of the brain. But as the animal goes around, what you begin to see is that there are areas of firing and there are areas there are patches of no firing. And then slowly what builds up is this representation which essentially builds up this very nice hexagonal pattern where the pattern can be described in terms of the spacing between the fields, and they can vary, as I'll show you in the next slide. The relationship between the major axes of the firing fields relative to the wall, and the relationship to each other. So you can see. After a bit of time, you can see that there's this very nice hexagonal pattern across the brain. One of the things that's been recently discovered, and I'm sure that a couple of weeks from now you'll have lots and lots of talks about the grid cells. I don't want to say too much about them, but I think one of the most important things that's now been discovered is that the different cells have different metrics, have different spacings between the peaks, and that this spacing is actually organized in terms of the anatomical relationship of the cell. So these cells are found here in the entorhinal cortex, and recently this effect was first noticed by Caswell Barry in our lab, but it recently has been shown very, very clearly and extensively by Stenstola and colleagues in the Moser lab, that if you record from different depths along the entorhinal cortex here, what you find is that the metric varies systematically depending on the depth of recording. So if you record from this dorsal position here, you find that the metric is very, very small, of the order of 30 centimeters or so. And as you go down along the dorsal to ventral axis of the entorhinal cortex here, you find that the metric changes, and it changes in terms of getting larger. So the spacing between the cells gets larger. It's almost as though you had placed a magnifying glass on these smaller ones, on the larger ones. And it turns out, according to them, and they have considerable amount of quantitative data, that the change is not a systematic one, but it's a quantized step, and it looks as though in any given animal there are really only about 4 different metrics, only 4 different spacings.
No, it doesn't look very quantized.
Huh?
Look very quantized at all. The first one's a continuum, more or less, right, with a couple peaks. Then they only have like a couple cells up there, so it can't really be.
This is just one animal. So across all the animals, I haven't put the data here. Edvard's coming, isn't he?
Yeah,
You can tackle Edvard. Yeah. We had much more limited data, and we certainly got the impression. Caswell Barry in our lab got the impression that it was fantastic.
Is there anything special about the ratio between those length scales?
Yes, 1.42, square root of 2. But that, I'm trying not to invite the next question, which is what the hell does that mean? So that's an average. It varies a bit from one animal to another, but it certainly is suggestive. It's suggestive that there is something, perhaps it's just, some mathematicians have suggested that this is the way to maximize your information about metric, is to have a system like this. It could be something which is just some biophysical thing, that whatever is producing this metric can only take certain, some particular values. Some people have seen it as evidence in favor of continuous attractor models of these. Wow. Should I answer? Some people have seen it as evidence in terms of continuous attractor models, in the sense that you could have totally independent models, but then you'd have some trouble as to why there would be this relationship. But given that you can have quantized oscillators, then it seems to me that it doesn't quite speak to those sorts of things.
It kind of looks like the Fibonacci series as well, like the spacing.
Yeah, it's a higher Fibonacci series, not the simple Fibonacci series. It's where if you got a rabbit which skips ones, yeah. And you can use these to actually, if you just add together grid cells at different scales, you can use them also and do a bit of thresholding. You can also use that to do these place cells. I just don't want to say much about it. I'll say a little bit more on tomorrow. But if you look at a grid pattern like this, then one of the first things you begin to think about is whether you could decompose this into its Fourier spectral components. And this is exactly what Yulia Krupich in my lab did. So she said, well, why don't we just see whether these patterns could be broken up into numbers of bands which are orthogonal to each other. And in fact, it works very well. This originally came out of some ideas that Neil Burgess and Caswell Barry and I suggested about the way in which you could build up the grid patterns using interference patterns between oscillators. So you can actually represent, for example, this grid cell as the summation, or you can decompose it the other way around into its Fourier components. That suggests, if that's true, that it might be possible to find these Fourier components, or that you might see other patterns which were not grid cells, which would, in areas in this parahippocampal region, which were also summations of different Fourier patterns, but which are not the canonical grid patterns. And so what Yulia did was she looked at a whole set of cells which we call spatially non-periodic cells, in, again, now another one of these areas, this parasubicular area here, and found that, yes, indeed, there are spatially periodic cells which are not grid cells, but which also can be decomposed into their spatial, into their Fourier, two-dimensional Fourier components. So here's the standard grid cell with its very nice hexagonal pattern. But then there are these other kinds of cells here, which when you look at the measure of their gridness, i.e. how good the peaks fall into this canonical hexagonal pattern, what you see is that they're pretty lousy at doing that, whereas a canonical grid cell has a score of about 1.3. These are way down and wouldn't qualify for it. However, they're very stable, so they're repeatable from one trial to another in the same way that grids are. And some of them begin to look like they have this very, very stripy, they're not quite these stripy cells, but they have this very stripy firing pattern. And if you look hard, you can find several of these. So here are these band-like cells showing this very nice stripy pattern, and here's their autocorrelation showing a very, very nice stripy ones. Here are a couple more.
Do these cells perhaps sense the edge? You showed earlier that if they're edge-sensing cells, they might spread out.
So there are the ones that I showed you, which are some relating to the boundary. These are a different class of cell. Yeah. I'm not saying that they're not related to the edge in some way, and this is something which is really quite a hot topic now. I'm sure will come up later in the conference that both the grids and probably these cells as well are somehow or other respecting the boundaries and taking into account. But it's certainly not clear exactly how they're doing it, and it's still sort of a grabs. So this suggests, and I'll say a little bit more about it tomorrow, this suggests that maybe the grid cells are a special class at the end of a continuum of these periodic cells, spatially periodic cells, some of which, like the grid cells, actually have very nice, three very nice Fourier components at 60 degrees relative to each other, but others have different angles between the Fourier components, or might have fewer components, or in fact, one component. We have not seen pure band cells of the sort. They all seem to have these little gaps representing a second Fourier component. Okay, so I won't go on much longer. I'll just say a few more words about our recent work on using virtual reality, because it fits in with some of the stuff that Mayank made. So we know that there are these different kinds of cells, and the question is, for any given cell, what proportion of its firing, what proportion of its field, what is the contribution of these different inputs? How much of it is due to environmental inputs, and how much of it is due to this internal navigation system, this path integration system, which is actually using the animal's own internal movements, its own internally generated movements, its motor movements to update the cells. And for a long time, we've been trying to answer this question, and as you say, can you turn the lights out? We've tried that. It turns out, we put animals on running tracks, we've done all sorts of things. It turns out that it's really quite difficult experimentally to separate out as completely the environmental aspects from the animal's own behaviorally generated inputs. So, for example, if you put it on a running track, then that changes the stimulus things, because you're changing the animal's running, but you're also changing the stimulus environment. So recently, following work by Holscher originally in Tübingen and Chris Harvey in Tank's lab, we, along with many, many people now, have set up a virtual reality environment for our animals to see if we could get control over these variables. And this is the picture of the virtual reality as seen by the animal. I don't think this video is going to work. No. But essentially what happens is the animal runs on a styrofoam ball. You can tell as it's running, it rotates the ball. The ball is riding on air jets, a cushion of air provided by air jets. And you present the virtual reality environment to the animal, in this case on 2 video screens, but we've done it with 4 video screens, we've done it with domes, we've done it with, by we I mean Griffin Chen in collaboration with John King. And so here's the representation of the environment. It's a linear track, as you can see here. The animal runs from one end to another. It gets food through a small tube as it gets to the end. And we represent the cues in the environment. These large cues here at the end are on the side here by these symbols here.
Is the virtual reality meant to take care of those other sensory inputs, like smell?
Yes. So we originally, as the Tank, we followed him pretty faithfully. We just set up a visual world. But now we've incorporated olfaction into it, and once you've got it set up like this, and of course this animal, his head is fixed, so he's just sitting there. Once you've set that up, you can provide the rest of the sensory inputs that you want. So with all the information I'm going to tell you about in this first experiment that we've just published is on the basis of a visual world that we've created. So the first thing is that we find about as many place cells in this virtual world as we do in real environments. So which is, I guess I didn't quite expect that, but here, and once you get cells, it takes a little while for the animal to get the hang of the whole thing, but once he learns to run and he gets the idea of the game that he's playing, then you find cells and you can hold on to them for several days. So you can see here's a cell recorded for 3 days with a field right in the center of the track. The animal is running from left to right. And here's a cell recorded right at the start of the run on the track. And now you can ask the question, how much of the input to any given cell is provided by the sensory environment, the visual cues in this case, because that is the only external sensory environmental cues that he has, and how much is due to his own movements on the ball. And the answer is that a lot of the fields, 80% of the fields, are disrupted by cue removal. So here's an example. Here's a cell with a nice field right in the center here. You take away the cues and it stops firing. And interestingly enough, it's the cues on the side of the walls, not the cues that he's approaching or the cues that he's running away from, which provide the crucial information. You can confirm that by actually taking advantage of the fact that you can stop him from running on the ball, but have him move passively down the environment. You just drop the ball so that he can't move, and then you just move him. You move him in a yoked, controlled way, similar to the way he would have run through the environment, passively moving it, and you find that about a quarter of the fields actually respond during that passive movement through the field. So here's an example: fires, passive movement, and there's the baseline again. So about a quarter of the cells in this very, very restricted, confined situation are actually responding to just the visual stimuli. Now, we don't know, and I should flag this up, we don't know whether that's actually the stimuli as landmarks. We're assuming that it's probably stimuli as landmark, but we don't know whether it's landmarks or whether it's just optic flow or something like that. It turns out that movement is also very important, and that a large number of the cells change their location on the basis of manipulations of the relationship between the animals running on the ball and the amount that it moves in the environment. So given that we have control over all of this, we can actually change how far the animal goes for, say, 10 steps. We can change it from going halfway down the track to a quarter of the way down the track, like this. And that predicts that if the animal is doing something like, or the hippocampus is counting the number of steps, that taking it down the way, that it should in fact move the fields backwards against the running direction. And this is shown in this group of pictures here, where here's a cell which fires. He's now running in this direction. It fires about a third of the way down the track. Now at the half speed thing, what you find is that it fires, the field moves backwards along it, so that it's beginning to fire before the visual stimuli are telling it that he's in the right place. It turns out that the visual cues are also important, because if you take all the cues away, then the cell stops firing. And it's about, not quite a half, but quite a few, over 40% of the cells are actually using the information coming from the motor system. And finally, we can actually do that experiment that I suggested a long time ago to see whether, once the animal has localized itself, once the hippocampal cells know where the animal is, can they then use the path integration, internal information, to upgrade or update its location? And you can do that in virtual reality quite easily. You can put the animal down in the start, let it localize itself on the basis of the cues that are there, and then as it begins to run, turn off all the cues, so that if the cell is going to fire in the right place, it has to use some information about its own movement. It needs that information at the beginning, because if you take away all the cues altogether and don't give the animal any cues, then the cell doesn't fire in the right place. But if you do that, put the animal down, allow it to localize itself, and then turn off the lights as it starts to run, what you see is that some of the cells, in fact it's quite a few of the cells, about half of them, can actually fire in the appropriate place, suggesting that there is a kind of a relationship between the two types of input, such that the animal needs the sensory input from the environment. Once the system actually knows or can say where the animal is, it can then use its own path integration, its own internal information to update the representation. We know, of course, that path integration, as I'm sure Kechen will tell you, is only good for a short period of time. The errors tend to accumulate over time, so that you can get this impression of a system in which you find out where you are looking at the cues, then you can sort of integrate. Then you need to look again, because you need to confirm that the integrator is reliable and not going out of whack. Okay, I think that's all I'm going to say. I was going to tell you a little bit about some of the work that we've done on human, but I think it's probably a good place to stop. So to summarize, the hippocampal formation provides a cognitive map of a familiar environment, which can be used to identify the animal's current location and navigate from one place to another. I think the idea that there are two independent strategies for locating place, and of course nothing in the brain is really independent of anything else, but relatively independent strategies is a viable one, and there's quite a bit of good evidence to support that idea that one of them is based on the environmental landmarks, and the other is using one of these internal navigation systems which calculates directions and distances and uses that to update the representations. There's pretty good evidence now that the hippocampus in humans has a rather important role, although not exclusive role, in spatial navigation and spatial awareness and spatial recognition. But there are additional inputs, and Howard Eichenbaum will try to make the case that you can see these additional types of inputs in the rat in the rat model as well. Particularly information about time, and particularly information about linear time. So if you had a spatial system and you added a notion of linear time to it, that i.e. you time-stamped every visit to a particular location with if you provided a timestamp so that the animal knew that it had visited this location yesterday and not the day before that, that would give you the beginnings of an episodic or autobiographical memory system. So we can see ways in which, if it turns out that animals such as a rat have this temporal time-stamping mechanism, we haven't seen it yet, but Howard is going to make the case that they have, then we could even imagine that the rat had an episodic memory system. But if rats don't have that time-stamping, and it has to be a linear time sense, they don't have that time-stamping, then you can imagine that it's created somewhere by the time you get to primates or humans, so that you would have available not only the locations that the animal visited, but also the time in which it visited it. Let me just thank the people. We've had generous funding from lots of different government and non-government agencies, and I particularly point out these two, the Gatsby Charitable Foundation and the Wellcome Trust, who have provided us with a considerable amount of money to set up a new institute, the Sainsbury Wellcome Center for Neural Circuits and Behavior, which is going to be opening its door, and Peter Latham has snuck out, but he and the whole of the Gatsby Computational Unit will be moving to the center of the building, and we'll be bringing in quite a few neuroscientists, particularly systems and behavioral neuroscientists, when it opens in summer of 2014. Thank you for your attention.
So we have time for a few questions, please. John, you can field them.
Yep.
John, is it possible to leverage the virtual reality system to ask about a hierarchy of length scales on which you have to navigate? So, I mean, by that I mean that if I think about walking back to the West Cottages right now, since I just got here, I'm going to walk west, and then I'm going to have to navigate a more detailed, smaller area to get through the gate, to get to the right building, and then get smaller to get to my door. I mean, is it possible to do that now in the lab with the rats?
Yes, I think so. And in fact, I think it does open up an enormous range of experimental possibilities. I mean, I didn't say it at the time, but of course one of the great advantages of this model is that the animal has its heads fixed. So that means you can use two-photon imaging to record from large, particularly in the hippocampus, and you can record from large numbers of cells, several hundred cells at the same time in the hippocampus. That's large. And in fact, we've been able to do that already as the animal navigates in that environment. And we were finding that if you have about 120 cells, about the same number, about 40% of them have place fields in our environments. You can also patch the cells so that you can do intracellular recording if you can get stable enough conditions. But also, and I'm particularly interested in the sorts of environment, the manipulations that you can do to follow animals in that environment, if it understands it, right? The real problem is And to be honest, I was always rather skeptical that animals would understand. We have a lot of experience with virtual reality, creating virtual reality environments for humans,
Right.
and seeing how they navigate and whether it activates hippocampus in imaging experiments. But I really didn't think that rats would understand it. I thought, first of all, they're not going to like having their heads fixed, and it turns out that they don't. But mice don't mind it at all, so all those work is done on mice.
Mm-hmm.
But that they understand it quite well. They understand it very well, actually, surprisingly. But you're right. There's just so one of the things it enables you to do is to, when we were building this new building, I kept thinking, oh, we're going to need big environments labs. So the Moses have, they have this huge long corridor which they've got animals running from one end of the building to the other end of the building to try to get some idea of what happens in large spaces. So I was thinking, how are we going to build those sorts of things? And it turns out you don't need them, because in virtual reality you can have huge environments, and the animals do seem to understand that. So many of our environments now are very, very large and virtual environments. Mm-hmm. Yeah.
So once the animals get used, you said it takes a while for them to get used to the virtual reality environment, and then you see the emergence of these cells. But now once they are used to the environment, if you now change the environment, what is the time scale for plasticity, the plasticity required to remap the cells to adjust to the new environment?
Yeah, so we've done some of that. We haven't done it in virtual reality yet. And there's a whole phenomenon which you'll hear quite a bit about in the coming weeks called remapping. What happens, what's the relationship between the two environments? And in fact, the Moses have probably done one of the nicest experiments in which they can flip between the two environments on a very short time scale. And then what you see a kind of an oscillation between the representations of the two environments with a little bit of a time course and a little bit of decay between the representation of one and the other. And it looks as though that at certain points both environments are being represented at the same time. But in general, if you put the animal in a new environment, they acquire that representation very quickly, in a matter of minutes.
Quickly, minutes?
Yeah.
And
before it looks like the representation is stable.
And could you comment on what are the plasticity mechanisms that contribute to that?
Well, it looks as though, and I guess there's quite a bit of work on knockout animals and everything. I think still one of the best is work that was done in New York, in downstate, where they put animals into a new environment, and then after the representation had stabilized, as far as I could tell physiologically, they then injected an MDA blocker and showed that the representation was lost the next time you put the animal into the environment. So that it looks as though you need to have some process that takes place after the animals had the experience, perhaps during sleep, perhaps during some consolidation phase, which strengthens the representation, and that's based on the classical memory transmitter energy. Yeah.
It seems to me that there is an implicit assumption that some cells are more modulated by internal cues and some cells are more modulated by external cues.
Yes.
That these modulations affect them differently. Are these hardwired or?
Good question. So you're right, and there's a slide I have which sort of plots them against each other, plots all the data against each other, and you take your money, you pay your money, you take your choice. So you can have some cells which are completely controlled by the environmental cues, some which are very, very much, given the caveat that the animal has to know where it is, and cells have to know where they are to start with, very much based on the internal path integration. So, and the question as to why that is, we don't know. But we think that we can actually begin to address that using the two-photon microscopy. It could be slight differences in the anatomical inputs to the different cells. Although looking at CA1, it's going to be very hard to imagine that the anatomy will support that, although we have some ideas about how to do the experiments. Yeah. Hi, Stefan. Sitting back there quietly. I was going to tell you, Stefan was going to answer all your questions.
My question is, in the virtual reality, when you when the movement is only by showing the visual cues, is the theta oscillation affected by that manipulation?
Yep. So this is in the passive movement. Yep. Yep. So in general, the theta is weaker, the amplitude is smaller, and the frequency is lower. And every once in a while, it's my impression that you don't have much theta at all. So, and the problem with that is that we yet haven't worked out whether that passive movement is part of the path integration system or not. I like to think of it as demonstrating, or part of the demonstration, that it's the sensory cues and therefore the landmark pathway. But in fact, we haven't demonstrated that. We need to show that it's not optic flow. And so we're doing experiments to try to rule out the optic flow component of that. So if you're asking whether the theta system is very much tied to the path integration system, I think the answer is yes. But I don't think the evidence is hard enough yet to be sure of that. No. Okay.
Fields that were preserved when you turned out the lights in
Yeah.
the virtual reality.
In the virtual reality, yeah.
Yeah, so you concluded that they were kind of distance indicating cells, but might they be time cells? Like were the trajectories sufficiently stereotyped?
Yeah, and I think Howard will talk about this quite a bit. The whole point about a spatial system, the whole nervous system works on the basis of temporal signals. That's what you got. You got cells firing, and they fire relative to each other, and so on and so forth. So the trick has to be to get a spatial system from that, is you have to take into account things like the animal's velocity, and you have to neutralize all those sorts of things. And it's the same thing with time. You have to, you can't use time to use space, or at least I can't see how you can do it without taking into account the animal's behavior. So, on the other hand, you could start with a temporal signal and then begin to use other types of information to, as it were, correct that. So how long have I been walking? Well, if I have no other information, in other words, if the only measure I have, or the only information I have about how far I've gone is how long I've been walking, well, that's better than nothing. So you might start with that, but then you would have to modulate that by things like, am I walking fast, am I walking slow, is the wind in my face, is it behind me, all those sorts of things. But if you got rid of all those other things, then you might end up with what looks like a pure temporal signal. So I think that's a possibility, and I wouldn't want to rule it out, and I think there's some evidence, even before Howard's evidence, that that the theta rhythm can be used to measure time in some of these conditioning experiments. So I wouldn't want to rule it out. I think it's not going to be a very good measure of distance. So it would also have to be integrated if you want it to be a pure temporal signal, i.e. to produce this spatio-temporal system, which we'd all love to believe that they're right, but which I'm afraid I still don't see very much evidence. Yeah.
So you know that we looked at this place cells in virtual reality too.
Yeah. Yeah.
And we found very profound impact on theta rhythms, speed dependence of frequency.
Mm.
Do you see any of that?
Speed dependence of
frequency of theta. So frequency of theta rhythm very clearly depends on the running speed of animal in the real world, which is kind of used for a variety of theories.
Yes.
Whereas in virtual reality, that was completely abolished.
It was pretty much. It was abolished.
Pretty close to zero. That is as much, pretty much, as you can imagine in biology. And
so we haven't. So that's a good point. We haven't looked in the virtual reality on speed dependence.
And then the other.
But there's certainly, as you say, in animals running around in the real world, there's a very strong dependence. The frequency is dependent on the speed, which is part of kind of this idea that the theta system is part of the system which is taking or providing some of the information about how fast the animal is running by modulating the frequency of the theta.
So why do you think it's gone in the virtual reality?
So when we looked at in the virtual reality, the theta rhythm has a certain frequency, 8 hertz.
Right.
That frequency increases with the running speed
right.
in the real world. In the virtual world, that's abolished, pretty much gone. It's almost all or none.
Yeah, and you also get the frequency is lower, don't you?
Frequency is slightly lower. That's right.
So the problem is that there are lots of it's complicated. How about that? There's the answer. It's complicated.
So, but why?
So there's only one. So there's one component of the theta rhythm is shows this very nice relationship between theta frequency and animal speed. But there is a baseline level at which that relationship disappears. It's kind of sort of the zero crossing of the plot, and that looks like it is a rather fundamental aspect of the theta, which is basically in the old literature, Case van der Wolf used to talk about that as acetylcholine sensitive. So we would say yes, if there is a loss of the frequency, and we've shown this pharmacologically, we showed it, Colin Weaver has shown it, that component, the non-frequency dependent component is cholinergically based.
Could that be due to lack of vestibular cues?
Well, that's a very good question, and that was another reason I thought that thing wouldn't work at all. So this animal has his head fixed, so he has no vestibular information at all, and he can't use that information. And certainly, if you put animals on rotating tables, and Kay has also looked at this and points about the relationship of the vestibular input by putting animals on slowly or rapidly rotating tables. It certainly is part of the driver for that whole theta oscillatory. So I think one shouldn't. I'm amazed at how good the system works, but one shouldn't fool oneself that this is the whole world. And it's certainly we've lost a lot of things. We've lost all the sensory inputs we don't know about, but we certainly lost this.
So
there was a question up there. Yeah, okay. Yeah. Did you have a question? No. Oh, okay. Sorry.
So in this virtual world, would you say that these visual cues, landmarks, they are sufficient to get spatial selectivity?
Yes. Well, that's I think what we're seeing. In a quarter of the cells, it looks as though whether if you gave the animal nothing, but in other words, if the animal, if you just passively moved it around, then would you get it? I doubt it.
No, just
but once he's been moved around and has been able to integrate the two inputs, then it certainly is the case that a lot of the cells, a quarter of the cells, are actually activated purely by the visual inputs. And I would guess that when we produce these complex multi-sensory inputs, multi-sensory worlds, that we'll get even more of them being activated.
Have you ever looked at two rats in a maze? Does a rat perhaps also have a relative system where he checks how close or how far the other rat is from me, and where is he located respectively? Yeah. You could imagine a similar place in grid cell system which is relative to the position of the two animals.
Yeah, so that's a good point, and of course the system we're describing is an environmentally based system. That's not to say that there aren't lots of other systems which are based on frameworks centered on the head, as the animal moves around, things would change, but it's centered on other animals. But they're probably not hippocampal. There's no evidence that they're hippocampal. And I think all of us have, every once in a while, when we got frustrated, have thrown another rat in with the. Let's see what happens, and the answer is usually nothing. And it's now been formally done, and there's no evidence at all that other rats provide much.
Because humans are very good in...
We're very good, and I'm sure that the system is powerful, but I think it's nothing to do with
somewhere else.
It's somewhere else.
But related to that, we see these distant cells in virtual reality instead of place cells. Do you see that too?
No.
So you have head rats run back and forth, the mice run back and forth on linear tracks?
Yes, and they have...
And you see place selectivity, not distant selectivity?
Yes. Yeah. No, our results are very different from yours.
Did you have quantification of place versus distant selectivity?
Yeah, no, it's place, yeah. Huh. And they fire in one direction.
No, we do see cells which are directional.
Yeah.
But then if you look at cells which are bidirectional, which means cells that fire in both directions, then we see pretty much all the cells, they fire at the same distance and almost never at the same place.
No.
Have you looked at those
Yeah, we haven't seen
bidirectional
that.
cells?
Yeah, no, we don't see that.
You looked at it and you didn't see them.
No. So there's something different about our virtual reality environments. We get it.
What's the difference? Distance measured from where? What is the
Along the
difference?
track.
It's just distance along the
If
track.
I start here, the distance defines the place. So what?
No.
I'm not understanding the distinction.
No, it's different. So for example, what we found is, let's say the rat is running from here to there and back. Then in the real world, if a cell fired here when he was going from here to there, when he goes back, the cell fires at the same place. It's a place cell.
So those are,
But let me.
so those are cells which are probably, they're almost
what we
entirely.
find in virtual world is that never happens. If a cell fired when he left from here at this distance, it'll fire at that distance when he leaves from the other side. So it's entirely the opposite.
Okay.
And we are wondering if you see that too, and you
No,
don't
we
see
don't.
it.
No. We see the same things in the virtual reality as we see on the normal linear track in the real world. Yeah.
Can you tell us something or speculate about the extent to which visual inputs to place cells are dependent on the grid cell representation or are a separate?
So I think they are probably separate. So we think that information is coming into those boundary cells, which are found, which are independent of the grid cells. They're found in similar places, and all of them are projecting into the hippocampus. But at this stage, we think they're reasonably independent.
So then the remapping that you see in grid cell, you think is a consequence of what's happening first in the hippocampus, or are grid cells receiving another set?
Very good. Yes. Well, so I ducked the question. What's the relationship between grid cells and place cells? Moses will tell you that grid cells have taken over all of the world and are responsible for everything we thought the place cells were responsible for. And I don't think that's true. So it's a kind of an interacting system, and you need, and if anything, to be honest, you need the place cells and certainly Stefan will provide. You're talking at some point, are you? You're not just here to listen. You'll provide evidence that you need, or at least that things which disrupt the theta activity will disrupt the grid cells, but not the place cells. And there's other evidence, I didn't mention it, but our developmental work, what we would see when the cells development, suggests that the place cells are there in young animals before the grid cells. That's not to say that as I said, that the place cells can't use that grid cell input. So you shouldn't think of it as a linear system. It's a system which is it's a cooperative system.
Very good. Thanks so much.
Thank you.
So we have a break and we meet again at 3:30.